Welcome back. Today we'll be discussing how DNA gets copied exactly and why that's accuracy for life. Before we jump into the details, let's ask the big question. Why does DNA need to be replicated exactly? Well, each living creature has a unique genetic code carrying instructions for its own production. Wombats have different instructions than koalas, dogs, penguins, or mushrooms. Parents give rise to the, Parents give rise to offspring of the same species with similar genetic makeup so they can survive in the same environment. When scientists started searching for the code of life, they knew chromosomes are made of DNA and protein, so the genetic code that had to be made in one of these. Most scientists thought it would be proteins because they're complex and made of at least 20 different amino acids. But in 1953, Watson and Crick discovered the structure of DNA and found that genetic information is carried by a molecule with an alphabet of only four letters. Inside DNA just consists of an order of sequences of A, C, G, and T, also known as adenine, cytosine, guanine, and thymine. Before we jump into the details, cells need to divide for growth, repair, and reproduction, but how do we ensure every single cell gets an exact copy of genetic instructions? There are two types of cell division, mitosis and meiosis. In unicellular organisms like protists, cell division by binary fission is common. One organism becomes two, no sex cells are involved. In multicellular organisms, however, mitosis leads to formation of two identical cells that contribute to growth. Meiosis is different. It gives rise to gametes that transmit genetic material from one generation to the next during sexual reproduction. We'll cover meiosis in detail later, but for now, remember mitosis for growth and repair, meiosis for making sex cells. Let's focus a bit on mitosis first. Mitosis plays four important roles. First, growth of multicellular organisms from the fertilized egg to embryo, infant to adult. Growth relies on mitotic division followed by cell enlargement and differentiation. Second, repair of damaged tissue and replacement of worn out cells. Have you ever wondered how your paper cuts, knife cuts heal? It's all mitosis. Now, thirdly, asexual reproduction. Like growing plants from cuttings or cloning organisms, mitosis plays an extremely big role in basically replicating the cutting or cloning organisms. Fourthly, and most importantly, genetic stability. Mitosis ensures precise and equal distribution of chromosomes to each single daughter nucleus. All resulting cells have the same chromosome number and genetic information as the parent. Now, here's something cool. Most organisms start as a single fertilized egg or zygote. You probably knew that. But inside that fertilized egg or zygote, the reason that we grow up into full adults, there are embryonic cells that divide repeatedly and they are also pluripotent, meaning they have a potential to develop into any tissue type. These are called embryonic stem cells. In mature organisms like us, when we're 18, 20, not all cells continue dividing. Dividing cells remain at specific locations like basal layer of skin replacing dead surface cells or bone marrow producing blood cells. Adult stem cells are pre-specialized. Brain stem cells only make brain tissue. Heart stem cells only make cardiac tissue, which is heart tissue. In plants, tissue that can divide is called meristem, found at the stem, root, and tips. Now, let's just have a quick overview of meiosis. Meiosis occurs in sexual reproductive organs and results in the formation of gametes. During sexual reproduction, two parents pass genetic material to the offspring. Now, to prevent the chromosome number from doubling each generation, because let's say a parent generation 1 starts off with 46 chromosomes. After sexual reproduction, the child would have 92 chromosomes if the both chromosomes combined. But however, meiosis prevents this. To prevent them from doubling, each parent must contribute only half their chromosomes. That's how the cycle maintains 23 from the mother, 23 from the father, mixed to become 46. Now, when that offspring has its own offspring, that he will give half of his 23, the mother will give half of his 23, then they will combine to form 46 for the new offspring. Now, that's meiosis, reduction division. When a cell divides by meiosis, DNA replicates first, then the cell undergoes two successive divisions. Meiosis 1 is where diploid cells, 46 chromosomes, divides into two haploid cells, 23 chromosomes each. Chromosome number halves, then meiosis 2, where those two cells divide again, that results in four haploid daughter cells called a tetrad. These gametes are vehicles of inheritance, basically, because they carry genes from one generation to the other. Meiosis also introduces genetic variation. We'll cover that more in chapter 5. Now let's hop on back to DNA. In multicellular organisms, all cells must contain the same genetic code so they function in a controlled, coordinated way. Along chromosomes are long sequence of base pairs forming units called genes. Genes code for protein production, which makes up cell structure and regulates cell functioning. Bones, hair, skin, muscles, connective tissue all contain proteins coded by genes. Now, enzymes are proteins controlling all cell functions: respiration, photosynthesis, every biochemical reaction. It's vital for cell functioning and species stability that genetic code passes on without error. During mitosis, DNA replicates precisely and exactly, and copies are divided equally. Cells, well, now they're clones. DNA codes for proteins using inside cells and exported proteins like hormones coordinating other cells. Now, a change in DNA, also known as a mutation, may change the end product and harm the cell by interfering with structure or functioning. Sometimes mutations have no effect, but occasionally, sometimes they're also beneficial. Now, let's jump onto the cell cycle. Now, the cell cycle has four main phases. By the way, if you're confused, this cell cycle includes for both meiosis and mitosis, where the DNA is basically replicating. Now, the four main phases. The first phase is G1, which is a gap phase for cell growth before the DNA replication. Now, the cell becomes massive here. Enlargement occurs slowly, but metabolic changes prepare for division. Then the cell commits to the division. Now, the next stage in this is S stage, where basically the synthesis phase, where DNA replicates. DNA is copied, so each dividing cell has two identical copies at the start of mitosis. Now, we will go through very thoroughly on how DNA replicates towards the end of the video. G2 is the second gap phase after replication where enzymes check duplicated chromosomes for errors and correct them, and cytoplasmic materials accumulate for division. Then mitosis, divisions of nucleus occurs, followed by cytokinesis, division of the cytoplasm basically. Now, the cytokinesis marks separation of one cell into two in actively dividing tissue. After cytokinesis, however, each single cell enters G1 again to repeat the cycle. This begins with assimilation and enlargement, where cells increase size by assimilating nutrients. End products of digestion in animals, photosynthesis in plants. Now, not all cells continue cyclical division. Cells that need frequent replacement, like skin cells and digestive tract lining, divide rapidly. Some adult cells don't divide frequently, others, like nerve cells, may last a lifetime. They are terminally differentiated in the G0 phase and only re enter under special circumstances. Before we dive into mitosis stages, let's understand chromosome structure. In non dividing cells, DNA wound around, like, they roll up themselves around protein. It's called a chromatin. Chromosomes contain linear sequences of genes, units of heredity coding for inherited characteristics like eye color, hair color, height. Now, each organism has a set number of chromosomes. Humans have 46, like I said before. Platypuses have 52. Even lettuce, they have 18. It's all set for the species. During S phase, chromosomes replicate and the two copies of chromatids are held together by a centromere. These sister chromatids, because they're two chromatids, you get it, sister chromatids, separate during mitosis and move to opposite poles. They're now called daughter chromosomes and distribute into two daughter nuclei. Then cytoplasm divides, separating the nuclei. That's cytokinesis. Now let's look at it a bit more deeper. Now, mitosis is continuous, but it's easier to understand in identifiable stages. There are five main stages. Interphase, during which the DNA replication occurs, like I told you, SG1, G1S, and then G2. Then prophase, metaphase, anaphase, and telophase. We'll look at this thoroughly. Late stages are accompanied by the start of cytokinesis, basically where I said the cytoplasm starts to split. At the start of mitosis, however, DNA is so condensed that chromosomes are visible with a light microscope. To keep diagrams simple, I'll show you only two pairs of chromosomes, four total in each cell. Now, interphase occurs during S phase, where DNA synthesis happens. DNA replicates, making an identical copy. It appears diffuse and spread out as a chromatin, not yet recognizable as individual chromosomes. However, in early prophase, DNA begins separating into chromosomes. In prophase proper, I guess you could say that chromatin material shortens and thickens by coiling, and DNA separates into chromosomes now visible with a light microscope. Each chromosome contains two copies of DNA, and each copy is a sister chromatid joined by a single centromere. The nuclear membrane begins breaking down and basically disappears. Now, a spindle begins forming from broken down material and extends across the cell like imaginary longitude lines on a globe. Now, let's start with the first phase, metaphase. Chromosomes line up across the center or equator of the cell. Each of those chromosomes are attached to a spindle fiber by its centromere. Now, each chromosomes in this consists of two identical sister chromatids. Anaphase begins when proteins in the centromere are cleaved, allowing the sister chromatids of the chromosome to separate. Now each chromatid becomes it basically becomes a chromosome. Spindle fibers contract, chromosomes are pulled by the centromere to opposite ends of the cell. As sister chromatids separate, they're now called daughter chromosomes, pulled towards opposite poles with movement assisted by the centromere. So basically, what is happening is when the chromosome is in the middle, when before the spindle fibers are pulled, they are called a chromosome. When they're pulling during that process, we name it sister chromatids. But now, when they're at the end of the cell, it's become 46 chromosomes again. Now, that single chromosome will be called chromosomes again. Now let's move on to telophase. Telophase daughter chromosomes gather at opposite poles. The spindle fibers break down, the nuclear membrane and nucleus reappear. Now, when the nuclear membrane and nucleus reappear, the nuclear division or mitosis is basically now complete. This results in two different nuclei with chromosomes identical to each other and the original parent. See how it became 46 chromosomes? The chromosomes doubled in interphase, and now when we are finally in telophase, this results in two identical chromosomes, which are exactly identical to the original parent. Now, before it is completely done, cytokinesis divides the cytoplasm, which separates the two daughter nuclei so each is in its own cell. It, it's kind of different in plant and animal cells. Basically, in animal cells, cytoplasm constricts in the center between the two nuclei and pinches off, kind of like a balloon in the middle until edges meet, then twisting it into two different bubbles, like clowns do. However, in plant cells, a cell plate forms while nucleus is still in telophase. Thickenings appear on spindle fibers at the equator and join up, forming a cell plate made of pectin compounds. Cellulose is then deposited on either side, forming a cell wall to separate the two daughter nuclei. The outcome at end of mitosis and cytokinesis is basically two daughter cells with chromosomes identical to each other and the original parent. They have the exact function, and now the daughter cells then enlarge until the same size as the original adult cell during G1, and this cycle keeps on going, and that's pretty much it. Now, for the other half of this video, we are now going to look at DNA structure and how it has to exactly replicate, and most importantly, why it has to exactly replicate. Now, let's look a bit more at the beginning of the video, what we mentioned, Watson and Crick's model. This model showed DNA as a large linear molecule arranged as double helix, or like a ladder but twisted, with four nitrogenous bases which are held by hydrogen bonds. Their model suggested that genetic information could be stored in the order or sequence of bases making up the middle of DNA. But Crick realized that each DNA strand, a sugar phosphate backbone, is anti-parallel to the other. That means you have the sugar phosphate parallel on the sides, basically the ladder handles, and in the middle, the rungs of the ladder are basically the four nitrogenous bases: adenine and thymine, cytosine and guanine, basically. Now, each base has one end attached to the backbone and the other end hydrogen bond to connected to another base. Bases line up between backbones like ladder rungs, like I said. And Watson and Crick realized in the model to ensure backbones remain equidistant, a double ring purine base needs to bond with a single ring pyrimidine base. Purine adenine must bond with the pyrimidine thymine and A and T, basically. Now, purine guanine must bond with pyrimidine cytosine. Basically, pyrimidines are cytosines and thymines, and purines are sorry, adenines and guanines. They also predicted DNA strands would be held by weak hydrogen bonds between pair bases, double bond between A and T, triple bond between G and C. This fitted with Chargaff's Purdy rule, basically. In DNA, ratio of A and T and C and G is always 1. Watson and Crick realized this complementary base pairing could predict how DNA could be copied. They basically published a paper called Molecular Structure of Nucleic Acids in 1953, stating that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Now, Watson and Crick's model showed all requirements of hereditary material. First, DNA can carry in coded form all instructions for cell formation and functioning, despite having an alphabet of only 4 bases. Second, DNA structure allows its own replication. To copy DNA, each strand can serve as a template for enzymes to synthesize a new complementary strand. Third, DNA can be transferred from one generation to the next, packaged as a chromosomes and carried by gametes. Proteins associated with DNA, however, in chromosomes keep DNA neatly packaged. So, like I said before, the DNA coils around these proteins called histones, then, like sort of like thread wounding around a cotton reel. This holds long DNA threads compactly, so DNA is easy to sort and separate during cell division, and efficiently transported between generations. Histones also play a role in exposing DNA sections so genes can be expressed. Now let's have a look at what RNA plays a role in this. Basically, DNA stores genetic information controlling the cell and the whole organism. DNA is the main chemical in chromatin in the nucleus, with small amounts in mitochondria and chloroplast, basically. The thing is, DNA transmits inherited information from cell to cell during division and generation to generation during reproduction. But however, RNA is a nucleic acid in small amounts in the nucleus, larger amounts in the cytoplasm, however, usually associated with ribosomes. RNA have the base uracil instead of thymine. It has the same A, C, and G, but however, instead of thymine, it's basically uracil. There are 3 types of RNA: messenger RNA or mRNA. Now, DNA replication is production of 2 identical double-stranded DNA molecules from one original double helix. DNA replicates during interphase prior to mitosis, like I told you before, and each cell receives one exact copy of coded instructions. Watson and Crick noted their model suggested a possible copying mechanism. If strands separated, each could act as a template for synthesis of a new completely different complementary strand. This is indeed what happens. The process is called semi-conservative because each resulting DNA molecule has one new strand and one old or conserved strand. This mechanism should ensure genetic material is copied exactly. However, DNA replication isn't as quite simple as it appears. DNA has to unwound from its spiral before strands can separate. A large number of reactions takes place simultaneously, so a collection of enzymes is required. These 2 strands run in opposite directions, anti-parallel, and nucleotides can be added in one direction only onto the free 3-inch end. Replication errors sometimes occur and need to be corrected. Now, DNA replication occurs in 4 main steps. The step 1 is basically DNA double helix unwinds. Each DNA molecule is double-stranded helix. An enzyme called helicase causes the DNA helix to progressively unwind and start to separate a lot of helixes. Step 2, the DNA unzips. The 2 strands separate using ATP as energy. Helicase disrupts weak hydrogen bonds between complementary bases. Now the DNA strands separate, exposing nucleotide bases. If you think of DNA as a ladder, like I told you, each rung splits down the middle, starting at the bottom and creating a replication fork further up where the DNA is still joined. Now, over here in the diagram, as you can see, a replication fork is a point where the DNA is split. Now, wherever the DNA is split, it forms a Y shape. And around this Y shape, the helicase starts moving to the left and starts moving to the right in opposite direction of that replication fork. Now, for synthesis to be initiated, a short strand of RNA, a primer, is made of enzyme primase and attaches to it the DNA. Next, enzyme DNA polymerase III adds DNA nucleotides to continue synthesis of the new strand. It picks up free nucleotide units floating in the liquid background of the nucleus and inserts them opposite their complementary base partner, using existing strands as templates and keeping nucleotide pairing specific. Like I said, adenine always pairs with thymine, guanine always pairs with cytosine. The 2 strands in DNA run in opposite directions, anti-parallel. Each DNA strand has 3' end and a 5' end. Nucleotides are always added from the 3' end, like I said. Therefore, nucleotide insertion during replication is also anti-parallel along the two template strands. Now, the final base pairing is checked by another DNA polymerase enzyme, which recognizes base pairing errors and carries out base mismatch repairs to ensure the accuracy. Despite these checking mechanisms, a small number of errors still occur. About 1 in every 10 billion base pairs. Incorrect base pairing results in a change of DNA base sequence known as a mutation. Cells may delay progression of cell cycle until DNA repair during due to phase is complete. Each resulting DNA molecule, however, contains one strand of existing DNA molecule and a newly synthesized strand. So this is how basically DNA is replicated. But however, the replicated DNA molecules rewind into double helix conformation. The end result, like I said, is two DNA molecules, each a double-stranded helix identical to each other and to the original molecule. Now, replication of DNA is a complex process, and each step is controlled by one or more enzymes. Now, the thing is that helicase follows topoisomerase and unwinds double helix by breaking hydrogen bonds between bases, causing two strands to separate and creating a replication fork. Now, here's the problem. The DNA polymerase cannot start from scratch. It can only add nucleotides to existing strands. Now, the solutions for this is basically just RNA primers, like I mentioned in the last one. Primase enzymes synthesize short RNA primers, about 10 nucleotides long, provides a 3' hydroxyl group attachment point. Then it just gives 10 nucleotides, and after that, DNA polymerase can now begin adding DNA nucleotides. It's because polymerase can't start from scratch when there's absolutely nothing. That's when the RNA primer comes in, it gives its 10 nucleotides long, attaches the base, and now this allows the DNA polymerase to continue adding DNA nucleotides. Now, after this, DNA polymerase third synthesizes new DNA strands using existing strands as template. Nucleotides are added to growing strand from the 3' end. This joins phosphate groups of each nucleotide to previous ones, creating phosphodiester bonds between these molecules to make sugar phosphate backbone. DNA polymerase 1 is mainly functional in editing, recognizes and repairs base pairing errors. Now, let's just sum up. Helicase unwinds the parental double helix. Binding protein stabilizes the strands. Primase adds a short primer to the template strand, which helps polymerase continue the replicating of DNA. Now, DNA polymerase binds the nucleus to form new strands. Exonuclease removes the RNA primer and inserts the correct bases. And ligase joins the Okazaki fragments and seals nicks in the sugar phosphate bond. Now, let's just hop on to the primer removal and joining. Now, DNA polymerase 1, it removes the RNA primers. It replaces, because you can't have RNA, now that successfully that the DNA has been replicated, you can't have RNA primers laying around. Now, the DNA polymerase 1, this replaces them with the DNA nucleotides. It ensures entire strand is DNA, no RNA contained. Finally, the two new strands are sealed together by the enzyme ligase. The final base pairing is checked by another DNA polymerase enzyme, which recognizes base pairing errors and carries out base mismatch repairs to ensure accuracy. Despite these checking mechanisms, a small number of errors still occur, about 1 in every 10 billion base pairs. Now, after it begins with one before the chances become 1 in 10 billion. Let's just sum up. It starts from 1 every 30,000 errors per division. That is way too incorrect. Now, the layer 1 DNA polymerase proofreading, this makes it 1 in 10 million. Starts from 3' to 5'. Now, layer 2 mismatch repair enzyme, it scans newly synthesized DNA, corrects errors DNA's polymers missed, and now finally it becomes 1 in 10 billion. Let's just go through. So, why is it important? Why is it important of exact replication? This ensures accuracy of the semi-conservative replication, and most importantly, when you pass on to the gametes, if there's an extra chromosome, this could cause Down syndrome, which is common. This is why it's very important that DNA is replicated exactly. Now, the DNA structure, the proteins determine everything. The four letters that make up entirely your lifestyle, it determines everything. That's why the importance of exact replication is vital. Mutations can disrupt the protein functions, which ruins cells' functions. This can cause genetic diseases, but most importantly, this is how cancer is formed. Thank you for listening to my video, guys. Hope you learned